Hello, welcome back. So this is the real deal. It can be very confusing to just read off what I've written here, but it's a lot of things, but we can get through it together. This is fatty acid oxidation. This is where beta oxidation takes place. This is where the body reaches out and says, okay, I need my second source of energy. I need to oxidize those fatty acids to get maximum energy out of them. And it occurs definitely in the liver, the heart, and the muscles. So just to have a general idea of what this process includes, it includes activation, it includes transport, and it includes beta oxidation. Okay, so fatty acids, just like any other molecule, can be transported into liver, and they may not be in their active form, and so their active form does not contain a CoA molecule. So an active and an inactive form would be taken into the liver and then converted into an active form by the effect of fatty acyl CoA synthase. So we're creating this active form, so that's why synthase, fatty acyl CoA synthase, and it's an energy generating step. ATP is used and CoA is added. Okay, remember, inactive, active, under the effect of fatty acyl CoA synthase, CoA is added, ATP is used, and then so fatty acyl CoA is the active form of the fatty acid chains that we will use. Now, when fatty acyl CoA is produced, it cannot be transported into the mitochondria. So what it does, what the liver does or the body does, is convert it to a different form that can be transported into the mitochondria. And this takes place by an enzyme called CAT. It says carotene, the full form is carotene acyl transferase number 1. And so now this enzyme converts fatty acyl CoA into fatty acyl carotene. Okay, and so this involves the removal of the CoA molecule. Now fatty acyl carotene is produced, it's transported into the mitochondria. Fatty acyl carotene is inside the mitochondria. Now we want to get back to our original molecule to be able for us to use it in the beta oxidation that will take place in a single bit. Now carnitine is this fatty acyl carnitine is acted upon by a second enzyme. Here it is called CAT2, which is carnitine acyl transferase number 2. Okay, and so this converts the fatty acyl carnitine into fatty acyl CoA back again by the release of carnitine. Carnitine is taken back outside for recycling and transporting of fatty acyl CoAs. When we have this molecule, fatty acyl CoA, now we are really ready to go into beta oxidation. To summarize what beta oxidation is, I have a 4 letters that you can keep in your mind. It says O hot, so oxidation. Okay, hydration, oxidation, and then we have the final enzyme called Thylaes. So just keep this one in your mind: oxidation, hydration, oxidation, Thylaes. Okay, so the first step is oxidation, and it takes place by the involvement of FAD. So FAD plus is converted into FADH2 under the effect of acyl CoA dehydrogenase. Okay, so FAD to FADH2 under the effect of acyl CoA dehydrogenase. Whenever you see an H2, of course, there's a dehydrogenase. Acyl CoA, we're just talking about the fatty acyl CoA, so acyl CoA dehydrogenase. That's the first one. First process gone. The second one we're going to talk about is the hydration. Hydration means the addition of H2O molecule, and this takes place in a bit. So this reaction converting fatty acyl CoA into the next product under the effect of acyl CoA dehydrogenase and FAD, the product here is enoyl CoA delta 2. So what we are doing here, where did we take those two H2s from? It's from here, from the alpha and beta carbons. Carbon number 1, carbon alpha, carbon beta. So we take H2 from those two, and so when we take H2s from H1 from here, H1 from here, two H2s, now double bond is formed between the two alpha and beta carbons, and this product is called enoyl CoA trans, because H is basically found like that and found like that on opposite sides, so that's why these H's appear like that, so that's why trans. Delta 2 is because it's between the alpha and the beta. So trans delta 2 enoyl CoA. Now the enzyme that does the hydration, the addition of the H2O, is called the enoyl CoA hydratase, which adds H2O to this molecule to give an OH to this beta carbon and an H to this alpha carbon, and then generate something called the beta hydroxy acyl CoA. Beta hydroxy acyl CoA. Okay, now beta hydroxy acyl CoA, as I said, beta has an H, OH, alpha has an H and an H. Now we are gonna go and so we've done hydration, and now we're gonna go into the second oxidation. And the second oxidation here, NAD is involved. NAD is converted to NADH. How can we know when does FAD or NAD come? Just remember, F stands for first, so it always comes first. So first oxidation FAD, second oxidation NAD. Okay, so now NAD plus is converted to NADH under the effect of beta keto acyl dehydrogenase, because what we produce in this reaction is beta keto acyl CoA. Why? Is because a ketone body is evolved at carbon at the beta carbon when the H's from the beta are removed. So now we are forming a ketone body right here. So now it's called beta keto acyl CoA. Beta as in the ketone is on the beta carbon, and acyl CoA, which is the product that we've been always using. And then finally we have the Thylaes. Thylaes effect causes the breakage of the bond between the alpha and the beta, and then the addition of an additional CoA onto this molecule. So now we have to know that when we add a CoA onto this molecule, we're adding it to this part right here that does not have a CoA. This part has a CoA. So the part to the left of the bond breakage does not have a CoA. The part to the right of the breakage has a CoA. And so this molecule right here just generates an acetyl CoA, which is shown right here. And this molecule on the left would generate another fatty acyl CoA, which will then be taken back and regenerated into this process again and again and again. Now, the beginning product that we had a 16C structure. This fatty acyl CoA has a 14C structure. Why? Is because we have removed two C's as a form of an acetyl CoA. And so the acetyl CoA that is removed will be used in the Krebs cycle to generate ATP, which is the general goal of our fatty acid beta oxidation. All right, so very can be very confusing, but it's also very simple. So now, just a general idea. So 16C fatty acids would generate about 8 acetyl CoA. So the number of acetyl CoAs produced from an even number fatty acid would be the number of carbons divided by 2, which is 8, and the number of beta oxidation rounds that take place from this molecule is basically we take 16 divided by 2 to give you the acyl CoAs, and then you subtract 1 to get the beta oxidation. So in this case, 16 divided by 8 divided by 2 is 8, minus 1 is 7, so 7 beta oxidation rounds. Now, the professor may or may not ask us about how much yield do we get from a specific even number fatty acid oxidation process. Let's say now I'm going to take you into this calculation and show you how this is done. So you have a 16 carbon fatty acid oxidation. As we all know and discussed, we have 8 of them acetyl-CoA produced. So now we have 7 rounds of beta oxidation, and we're going to use this as a guiding process. Okay, so to figure out how many products of each is produced in the Krebs cycle from one acetyl-CoA. So in a Krebs cycle from one acetyl-CoA, we know that there is 3 NADHs produced, 1 FADH produced, and 1 ATP. So now if these are produced from one, how much of each are produced when there's 8 rounds? So we just multiply 8 by each number. 8 times 3, 24. 8 times 1, 8. 8 times 1, 8. So this is from 8 CoAs. But we have to always subtract 1 ATP because, as remember, we always use an ATP to activate a fatty acid in the beginning of the process. So keep those figures in mind. Now we have to figure out how many of these products are produced in the beta oxidation. So the Krebs cycle is basically this part right here. So you see, we generate acetyl-CoAs to go into the Krebs cycle, but we also generate products in the beta oxidations itself. So we have to know how much of each product is produced in the beta oxidation. So 7 rounds of beta oxidation would give us NADH 7. Why? Is because in the beta oxidation 1 round, we have only 1 NADH produced and also 1 FADH produced. So in 7 rounds, we have 7 times 1, 7 times 1, right? Or 7 times 1, 7 times 1. Gives us 7 NADHs and 7 FADHs. Now we just have to go and add those numbers onto what we have here. So now we will have NADH is 31 in total because 24 plus 7 is 31, and FADHs is 8 plus 7, 15, and ATP, as we know, 8 minus 1 is 7 because we're using 1 ATP in the beginning. So now we have to get the total amount produced in when it comes to ATP. So we know that when it comes to NADH, we know that 1 NADH produces either 2.5 or 3 ATP. If you're looking at literature, it may tell you 2.5. If you're looking at a different textbook or something, it may tell you 3. But this is the general value of how much ATP is produced from 1 NADH. And from 1 FADH is either 1.5 or rounded up to 2 ATP. So we take those numbers and multiply them by the numbers that we have. So NADH, we have 31. 31 times 3 would give us 93. 15 times 2 would give us 30, plus the 7 that we calculated originally. These are the number of ATPs that give us when we calculate the number of ATPs using the values 3 and 2. So in total, we will get 130 ATPs generated from a 16 carbon fatty acid compound. Imagine. And then that's just taking into consideration when you are saying NADHs give 3, FADHs give 2. And if you're looking at specific literature, they may tell you 2.5 is the value of NADHs or 1.5 is the value of FADHs. So these are ATPs, remember. ATPs. If you're looking at this, then the number would be 107. Okay, so this is how you would calculate the yield of fatty acid beta oxidation. Thank you guys for tuning in, and I'll see you guys in the next video.